Hello. Hi. Um, Hi. Thanks for the introduction, Erica. We are excited about the closing talk of the evening. Thanks for staying out so late. I am Suman, a postdoc in the Anderson Lab, and I have with me my partner in crime, Amanda, who is a graduate student. And together, we are very interested in cell-based therapies. So what we're going to tell you is a very short and sweet story about what is cell-based therapy, why are we excited, what it can do for us, and how we are actually in the lab solving the problem of translating this to the clinics. So, we are built of billions of cells, and all cells are programmed to do a specific function at different locations. One can be a kidney cell, one can be a liver cell. But with the advent of genetic engineering, now we have the power to transform and reprogram these cells to give it additional powers, to make them like supercells. And now these supercells can be in your body and do a number of functions like find diseases, look for biomarkers in your blood. If it finds a biomarker, it can then produce the drugs right there. Like now we spend billions of dollars doing it in factories. And if you put the cell strategically right at the tissue where it needs to be, it can give the drug at the tissue. No need to take injections, right? So now you will ask the question, well, if this is so great, why can't we just take the cells, inject in us, and solve the problem, right? So there is a big problem there. Say you take our supercell and introduce you to it to the bunch of cells in the body. It looks very different because now we have reprogrammed it. And there the immune system comes in because the immune system has been designed to recognize what is not us. So these immune cells get called in. They're not happy about this issue. So they start launching an attack on this supercell and actually kills it. So there goes all your plan. So that's where we come in. So we are engineers. So we were like, what could we do about this problem? And our solution was actually very simple. Why don't we build a house for this guy where it is safe and it can do his work? So this is a very simple concept of a device where, if you notice, there is a porous membrane, and that's really the key engineering component. Now, what the membrane does, it prevents the immune cells from getting to the cell that you have transplanted. So now, even if they try very hard, these cells, even the sneakiest cells, cannot get in. And our supercell can stay where it is and produce drugs whenever it finds something is fishy going on in our body. So now we'll switch gears from Cartoon Network to Discovery Channel, where Amanda will tell you the science behind how we are doing this in the lab.
All right, thanks, Suman. So I'm just going to tell you a little bit about how we actually make these devices. So as Suman mentioned, we have these porous membranes, and we're able to fuse them to basically what is a plastic chip. So we assemble it, and then we have a loading port through which we're able to load whatever our therapeutic cell type is. And then we can seal it. And here's an initial image showing the overall device. It's a top-down view. And here you can see we've loaded our therapeutic cells. So if we zoom in on the device and we take a view from the side, you can see that once it's assembled, you're able to load the cells, which are shown here in orange, into these channels. And some of the engineering aspects that we had here, the first is the pore size. So as Suman mentioned, this is really important for allowing diffusion of oxygen and nutrients in and also of the therapy out, but also for protecting it from immune cells. Another big thing that our lab has been looking at is different surface coatings. And so we've developed some coatings which basically protect our device so that the immune system doesn't recognize it as being foreign. And so the way that we took our images is that we are able to use confocal microscopy to look at a slice through the image. So it's basically a top-down view where we're able to see what the cells inside look like. So you'll see in the image on the right, which is similar to the one outside, that in black you can see the walls of the channels, and then the cells are within the channels. So in our final image, we'll see that we again have our therapeutic cells that are shown in blue. And with microscopy, we're able to monitor the cell behavior over time. And so in this particular case, we have, after the cells are able to mature, then they're shown in green. And then the mature cells are able to secrete therapeutic proteins, which are shown here in red, out of our device and into the body. And so we're hopeful that devices like this will be applicable to a number of diseases. So we're particularly interested in things like, for example, diabetes, where you have to do constant monitoring of your blood glucose level and injections with insulin. And also things that are chronic treatments and have systemic side effects, so things like cancer therapeutics. And so we're hopeful that the long-term goal could be that by looking at different kinds of patients, we're able to implant devices like these, and we can have these living drug factories for a variety of disease applications. And so with that, we want to thank our advisors, Bob and Dan, the Langer and Anderson labs, and a special thank you to the microscopy core in the Coke that we used to take this image. And thanks to all of you for listening tonight.